Okay, welcome back for the second part about immunity, the second video on innate immunity. And this is we're going to cover now areas of phagocytosis and how cells recognize these phagocytic cells recognize and basically help prevent an infection in the first line of defense against any potential infection or foreign invaders before they infect, as well as afterwards. This process is called phagocytosis, and basically there are two places you need to be concerned about being infected. One is in the blood and one is in the tissues, being infected in the tissues. In the blood, there's two types of leukocytes, granulocytes that are granulocyte and agranulocyte that are involved in phagocytosis. One are neutrophils, which are particularly important for antibacterial, as they have strong antibacterial activity, and they're about 60 to 70% of those types of leukocytes, white blood cells. And the other are monocytes, which are basically a blood-borne phagocyte, and they're about 3 to 8%. In the tissues, there are macrophages, microphages and macrophages. They're similar to neutrophils, and these are cells that also are the macrophages are against the early stages of infection. Macrophages are fixed in tissues. They're part of the what are called their reticuloendothelial system, or the RES system. They're active against microbes, and they clean up dead and dying macrophages, as well as dead tissue, until they actually die. So they have this kind of like it's kind of an interesting function. They function from everything, so sort of like the clean up the garbage of your cell debris that's around, as well as producing and converting those to nutrients, as well as first line soldiers with able to go after viruses and bacteria. The functions of phagocytosis are several. One is, as I mentioned just now, is to clear the body of debris, to take it in and get rid of it, so that it can be catabolized into new proteins and new nucleic acids and lipids. Secretion of defense proteins like interferons, as we've seen. Ingestion and killing of bacteria, so they have strong antibacterial effect. Inflammation, producing pyrogens that are able to go in, endogenous pyrogens, and activate the inflammatory response. Antigen processing, in which they're involved in, as we'll see later, we're going to see a lot of this later, they're involved in the adaptive immune response. So there's some crossover with not all phagocytes, but like, for example, with macrophages, and the adaptive immune response, as we'll see later. Now, how does phagocytosis occur? What we're going to give is just a general mechanism. A bacterium or particulate matter will bind to a potential receptor on the surface of the cell, and that bacterium is going to be endocytosed, and then it's then going to be endocytosed and contained in something called an endosome. These types of cells contain things, membrane-bound vesicles called lysosomes that contain enzymes and noxious potential chemicals, enzymes that produce chemicals that are destructive, as well as enzymes that can just go after and destroy bacterium. There's a fusion process that occurs here, and there's a primary forming the primary lysosome, and hydrolysis occurs and destruction of the bacteria into a variety of all of the various pieces as it's being dissolved, and this is referred to as the secondary lysosome. And then that goes and fuses to the membrane, and this is then excreted out of the phagocyte. So it goes through an endosome, it goes through endocytosis, and the endosome is formed, merger of the lysosome with the endosome to form the primary lysosome, hydrolysis, destruction of the bacteria, and then dumping out of the contents to the or catabolism contents to this to cell into the rest of the body. So here's a picture. Of course, phagocytes are not this. The phagocyte is this big red, hairy-looking molecule. Of course, phagocytes are not red. This is false coloring. Um, and it's in contact with bacterium, in this case E. coli. And you'll notice its surface is really has all of this structure. Has lots of its surface area has a very high surface area. It has all these structures that are continually sweeping and binding, potentially tasting and new things it runs into. And in this case, the bacterium, the E. coli, are these little green guys, these little green fellows. And there can be, and you can see they're orders of magnitude smaller than a bacteria, than a phagocyte, than a cell, um, than a human cell. In this case, the bacterium can be endocytosed individually, and you can see if we have a close-up, if we just close up of that activity, what we see is here's a bacterium. These are bacterium that are actually on their way into the macrophage. They're being endocytosed entirely into the macrophage to be destroyed. I have a little movie here, which I'll show you. What you see here is up at the top is a macrophage, and then at the bottom here, these two bright balls is Candida albicans type of yeast. And what you're going to see is it's going to move along here. It's going to recognize these, and then it's going to phagocytose them. Did you hear the sound it made? That's I don't know if you heard that sound. I'll play it again for you, so you can hear it. So that's what the sound of a macrophage eating a yeast. That's actually not true. And but it's just what you get the point across here is you can see um, it senses. See, it's sensing and engulfing. It's sensing and starting to engulf these bacteria, these yeast spores, and as it goes forward, they become taken up into it, and it completely engulfs them. And now it'll go through the process of destroying them. Now there are other things that the phagocytosis can do. In other things, it's not enzymes, but produce toxic molecules. Most of these have to do with molecules that are involved in oxygenation or reactive oxygen species. And so, for example, if you take glucose, it can be converted with NADP, but in the presence of 6G G6P dehydrogenase to pentose pentose phosphate NADPH, and that NADPH in the presence of oxygen and NADPH oxidase and cytochrome P can go on to form superoxide. The superoxide ions are not incredibly toxic themselves, but they can be in the presence of hydrogen peroxide, which can be produced. They can produce hydroxyl radicals, which are extremely toxic. They're very reactive and can react within one bond. They will react with and pull off a proton, as well as singlet oxygen, which is quite reactive. And so you have the presence of, whereas the superoxide dismutase actually gets rid of it, it also produces hydrogen peroxide that can combine with the other superoxide at seas to produce these very reactive oxygen species. Another one is the myeloperoxidase, which can reacts with hydrogen peroxide and chloride ion to produce a hypochlorous acid, which is basically bleach, and that's highly reactive, and can produce, and in the presence of water, can produce singlet oxygen. And those are highly reactive species that are very destructive by ripping off protons, breaking bonds, in particular with proteins and cells. So you have this production of a variety of different molecules. One of those, there are detoxifying reactions as well. Superoxide dismutase is one that can take superoxide to hydrogen peroxide, but again, hydrogen peroxide is reactive. Excuse me. The hydrogen peroxide can be detoxified by catalase, which is highly reactive to water and oxygen. So those are ways that the body has of also protecting itself against these reactive species. One other type of reactive species is nitric oxide. Nitric oxide is toxic and very reactive. It's a radical. It's a nitric oxide radical, and as you can see here, upon activation with interferon gamma leads to nitric oxide synthetase production, as well as another factor called TNF, tumor necrosis factor. And what I want to point out here is for the macrophage, in the presence of these kinds can be these kinds of factors which you see in inflammatory responses can activate nitric oxide synthetase to produce nitric oxide, which is quite toxic to cells, to infected cells in the vicinity of the macrophage, as well as any kind of bacterium in terms of destruction of those bacterium. So you see reactive oxygen species are being produced, nitric oxide, highly bleach essentially is being produced, things to detoxify and destroy the bacterium and infected tissues. Now the other part are macrophages express several receptors on their surface in order to recognize the bacterium. How are they going to recognize that bacterium? Why don't they just gobble up a normal cell, right? A normal human cell. Well, the way they do that is through, they have several types of receptors on their surface of their cell, which combine various types or components that you find on bacterial constituents. For example, there are ones that these are referred to as pattern recognition receptors, and we'll see about that. But there's a variety of these, as we see here: toll receptors, LPS receptors, scavenger receptors, mannose, and they will bind to a variety of these types of things, and those are the way that they will then take those things, those bacteria in by bacteria or the pieces of bacteria that they will bind to, that they recognize, and form the lysosome. In this case, it's referred to as the phagosome, phagolysosome, and primary lysosome, and the secondary, and destroy them. So the way that this all happens is referred to as pattern recognition. They're pattern recognition receptors called PRRs, and these receptors have specificity for pathogen molecules referred to as pathogen-associated molecular patterns. And so the receptors bind the patterns. So PRRs bind PAMPs. Pathogen recognition receptors recognize and bind to pathogen-associated molecular patterns. Okay, is that clear? These are the receptors up here, and these are the patterns. And so these particular patterns of things are what are recognized. And these are present on all cells of the same type. Binding to the pathogens results in an immediate response. So all macrophages contain the same kinds of these these types of receptors. Some of these pattern recognition receptors are referred to as toll-like receptors with specificity for pathogen molecules. They're referred to as toll-like because they are similar to a receptor that was originally discovered, I believe, in flies, which is referred to as toll, and these are like that, these are like that, and so that's always kind of stuck, and so for whoever discovered these, and so these are referred to as toll-like receptors, and there's a number of those who are that these toll-like receptors that can recognize different parts of bacterium. Now, you're not going to have to memorize all of these, but I do want to point out some of these that are different. Some of these are going to point out that are because they're relevant. They're quite relevant, and they're also quite relevant to potentially for development of drugs. There are some that will recognize diacylipopeptides. There are some that will in several different types, triacylipopeptides. Others that will represent like toll-like 5. That will represent that can bind to and recognize flagellin. Others that can recognize LPS, lipopolysaccharide, and they're given here in the sense that these are monomers here that bind. And when you see these two, they're crescent-like molecules, and these crescent-like molecules, in this case, form a heterodimer, TLR6, TLR2. Another heterodimer binds the triacylipopeptides, so it's TR1 and TR2. Some of these are a number of these are also found in the endosome when the bacterium or virus is taken up into the cell and is starting to be destroyed. You'll find them that recognize double-stranded RNA, that can recognize tRNAs, mRNAs that have folded mRNAs. They can recognize viral RNAs that are double-stranded. They'll recognize single-stranded RNA, and they also can recognize what are referred to as DNA, and they're referred to in this case CPGs. And these are really potent ones that can bind to TLR9, for example. CPGs are potent activators of the immune response. Here's one of those. Here's a crystal structure of one of those toll-like receptors. It's TLR1 and TR2, which is binding to the bacterial lipopolypeptide. And you can see here there has it's these two crescent shapes, and in the middle here in this space filling model is the molecule it's binding to. It's recognizing. Toll-like receptors are quite stable. They have this beta sheet structure. The beta sheet structure is typically very much stable. It's one of the most stable structures, as you probably learned in biochemistry. And so if you have something on the surface of cells, if it's a toll-like receptor, if it has a beta sheet structure, it will have a lot of stability. You can see here that the molecule binds across the two receptors. This heterodimeric pair, with part of the molecule binding in one and part of the molecule binding the other one. So the recognition of the molecule, you need both the heterodimer parts of the heterodimer to recognize it. The toll-like receptors all have this kind of crescent shape, followed by this, and then their ends they have these alpha helical structures. If we look at nucleic acids, for example, for double-stranded RNA binding, what we find here, what you can see here is that you see binding across the two. Now it's kind of across the two the two receptor molecules. In this case, this is a homodimer, and they're binding. And what you see is a homodimeric pair. They all bind with this homodimeric as these homodimers, and you can see here they bind. You can bind across. One binds on one side of the nucleic acid, the other binds on the other side. It's sort of like a sandwich, V-shaped sort of sandwich to bind the length of the nucleic acid, in this case double-stranded RNA, recognized by both of those strands, sort of put almost like a bar that a bar that links the two. Now there's a number of these that are important. I'd like you to know TLR9. I'd like you to know TLR TR9, and in particular because we'll see that's important for activation, and that isn't one of the more important ones recognized. But you see here there's a variety of these that are involved. They're all bind, and the receptors are, in this case, are all homodimers, and they bind to peptidoglycans, and you can see they have a variety of different things that they will bind to. They can TLR3 can actually bind DNA and RNA. And you can see here like TLR4 cells carrying it are macrophages, and the organisms that they come, these are the organisms that they come from as well. So, and then TLR7, which is a homodimer, has also been shown to be shown to be the target for antiviral drugs, the immunoquinolines. And we'll see that here. This is ubiquimod, which is a cream, Aldera, which is an effective antiviral for genital warts. It needs to be applied topically for HPV infections, and it activates the alpha interferon and other cytokine release. It activates that in terms of the macrophage are able to it activates those immune cells to do that, the macrophage to do that, and it has antiviral as well as has anti-tumor activity. This was discovered actually at 3M Pharmaceuticals right down the road from here, and you can see in fact that it has kind of a nucleic acid type of structure. This looks sort of like a base. You can see it sort of looks like a base of a nucleic acid. They're quite potent and activating the immune response. I had a student who was graduated from my lab, who went to work for 3M Pharmaceuticals, and he was actually working on projects to try to create more potent analogs that would be potentially more useful, particularly as anti-cancer agents, because if you activate the immune system, as we'll see kind of later, you can have not only antiviral but anti-cancer activity. And he made the mistake of one day when he was synthesizing in the lab of just touching his lip he thought his hands were clean, but his lip, apparently he had a little bit of one of those molecules on his finger. It touched his lip, and his basically the side of his face blew up. It swelled up because he had such a powerful immune response activation. And so these can be quite powerful, and they have to be used carefully. And you can see here, this is where Aldera is one of the places, is the place where it primarily binds, and its structure seems to reflect that. Now these toll-like receptors, as I said, as we have seen before in our principles, that you have a receptor, but it has to the signal needs to be communicated down into the cell. And you can see here, you're not going to be required to memorize this. I just wanted to show you that for this particular toll-like receptor that binds LPS, for example, TLR4 is a homodimer. It binds it and it leads to activation. This MyD88 is actually fairly important, but it activates and leads to then NF-kappa B activation, which we'll see later in terms of T cell activation. But these are transcription factors. There's basically signals that are sent, transcription factors are activated, and that leads to inflammatory cytokine production, such as interferon alpha, beta. And so that this is how this works. This is why it's so potent, because they lead to this. They have a signal that goes through and then activates those cytokines for release. So this is just another representation on, that you can see. These are the different kinds of things that bacteria that the macrophage can produce. These chemokines. We talked about interferon alpha. There's interleukin 1, interleukin 1 beta. The interleukins are all cytokine. These are really cytokines. They're peptides or small proteins that bind to cell receptors, and they cause a variety of biological reactions. These are their names. Uh, for example, IL-1 beta is involved in fever production of fever. TNF alpha is also involved in fever mobilization and metabolites, and can be involved in shock activation. IL-6 causes the activation of lymphocytes. It also involved in fever, and it can induce an acute phase of protein production by hemocytes. This IL-6 is proven to be really important one. And then there are other factors that we'll see later, particularly IL-12, which are involved in activating the adaptive immune response. So there's a whole range of signals and messages that are sent out by the cells. And in some cases, we're still learning how this whole chemical signaling is going on and being manifest, and what cells it's going to, and how those cells are being activated, the different cells that are being activated. It's very, very complex. If we look at again, take a step back and look a little bit more carefully at the physiological barrier, inflammation, which is referred to as a physiological barrier. These are the response to the damage of tissues by the microbial infection. Some of those things we've just seen: production of fever. These symptoms are redness, pain, heat, and swelling. And these all function to localize and to provide an environment in which will activate the cells, the macrophages and other cells, um, to really speed up their metabolism so that they can fight the infection. They also provide an environment which bacteria really don't like these high temperatures. And they also wall off. The swelling essentially walls off that area. It creates an area to sort of to localize the infection, to keep it from spreading. And pain and redness are part of that, to keep you from moving, to keep it localized. Now the fever is important. It's controlled by the hypothalamus. This elevated temperature is in response to, example, endotoxins, which are these bacterial lipopolysaccharides, as well as the pyrogens, such as IL-1, that are produced, which are endogenous. Higher temperatures, again, as like I said, speeds up the body's function and inhibits the proliferation of viruses, in particular viruses, and particularly rhinovirus, for example. You can tell if you have bronchitis or potential bronchitis, the difference between a cold and bronchitis, because a cold or rhinoviruses don't like to have a slightly lower temperature to propagate in, and that's why they stay in the head, because your head is actually a bit cooler than the rest of your trunk of your body, and so they don't migrate down or infect the lungs. Whereas with other viruses, maybe bronchitis, that way will infect the lungs, or adenoviruses, they will, and they can infect those lungs. And so viruses can have temperature can have a different effects on them. But if you raise that temperature, by and large, a higher temperature is not favorable for viral production, and that's one of the benefits of fever. Even though it's at the time you don't like it, it is the response that for controlling the viral infection. Inflammation has three major functions. It destroys to remove the injurious agent, the pathogen; confine what can't be destroyed, as I said, to wall it off, to create areas where it's walled off; and to repair that damaged tissue. Because the tissue gets repaired, the inflammation starts the process of destroying those areas that have been infected, and leading to provide some of the cytokines that are produced result in enhancing repair of that area and regeneration of the tissues there. There are bacteria can evolve faster than humans, and so they are able to come up with ways of evading the phagocytosis. And one of those is they may produce, once they've been phagocytosed, they may produce a toxin that will kill the phagocyte, or there's something about that's in their cell wall that's toxic that will kill the phagocyte. They can inhibit binding or endocytosis. They can maybe produce a protein, an enzyme that will destroy the receptor on the surface of the bacterium, or the surface of the cell, or destroy what's something that's marking it. They can overwhelm a phagocyte. They can maybe grow inside the phagocyte and just overwhelm the phagocyte. They're too big for the phagocyte. There's too many of them for the phagocyte to actually kill, and therefore they can have resistance to digestion in that primary lysosome and primary and secondary lysosome. And some of those, what you see is that's a hallmark, for example, of certain types of infectious agents like tuberculosis and leprosy and leishmania. These are bacteria that will mycobacterium that will grow inside the macrophage. The effect that you see from tuberculosis and leprosy, the effect is actually not the bacterium. It does it inadvertently, in the sense that it's a secondary effect. But a very primary effect is that the disfiguring and tissue destruction comes within basically a runaway inflammatory response in which the macrophages, which they're destroying the tissues, they're trying to save the tissues, and they end up becoming overactivated and destroying the tissues, destroying the tissues that you that is their hallmark, either in tuberculosis, the lung tissue, or in leprosy, the skin tissue, and area, yeah, areas of the body, like different types, like your different kinds of appendages and all. So we're going to stop here. Here's a review question, 3 review questions to take a look at. And why don't you stop? You can click and stop and answer these questions. We're finished here for innate immunity. It's a brief overview. There's a lot to innate immunity. There's a lot to inflammation. We'll be covering later in the course some other areas in more detail about inflammation in the microbiology section. So this will be the end of the video 2. I'm going to click and show you the answers, and then the second video will be finished. So I will see you later. I'll see you in class later.